students, welcome to Dr. B.R. Ambedkar Open University. Today I am going to teach you the part 2 of taxonomy of mulberry. So in this part 2 we will be studying about internal anatomy and structural overview. As mentioned in the earlier class, what is taxonomy? Taxonomy in Greek, it means taxo means arrangement and nomi, it means naming. That means arrangement on and also the naming. This taxonomy of mulberry, it gives us a knowledge about the classification, to which class or to which suborder or to which family does the species belongs to. Here in this mulberry we have seen that this mulberry relates and it comes under the family called as Muriaceae family, and its genus name is Morus. There are different types of mulberries that are present depending upon the climatical condition, depending upon the habitat, depending upon the soil condition. There are different types of mulberries that are present. There are I mentioned that there are Morus alba, Morus indica. These are there are different wild mulberry, white mulberry, black mulberry. Depending upon the habitat and also depending upon the soil conditions, there are different types of mulberries that are present. It is a wide range, a wide variety. As mentioned that silks will be majorly feeding on the mulberry leaf. As mentioned, as I'm telling you, mulberry, the silks will be majorly feeding on the mulberry leaves. As it has been feeding on the mulberry leaves, these mulberry leaves is high nutritive, it is having high protein, and it is having high carbohydrate. Internal, I mean the silkworm and the sericulture, it majorly focuses on the mulberry, as these silks will be feeding on the mulberry plant. So knowing and having knowledge of mulberry is very vital in the sericulture. Here the internal anatomy. In the earlier classes we have seen the macromorphology. Morphology is an external structures that have been present. Now we are seeing the internal anatomy. What is an internal anatomy? Internal anatomy is nothing but we are going to study the tissues that are present inside the body. Here, depending upon the roles, every tissue has its own role and the responsibilities to be done. So having knowledge also reflects the health and also reflects the quality of the cocoon and also reflects the quality of the silk. Here this mulberry is directly reflective on the quality and the quantity of the silk. Here we are studying the microscopic structures that are present in the stem, that are present in the leaf, and also that are present in overall plant. Here in this chapter we are going to study and we are going to decode the microscopic tissues which are present in the mulberry that will be helping for the growth and also for the development of the silk. This mulberry plant will be directly will be helping out in improvising the sericulture and the silk industry as well. In this lesson, we will be learning the anatomical mastery. That means we will be identifying the microscopic cell layer of roots, stems and leaves. All the tissues, all the microscopic structures that are present in the leaf, in the stem, and also in the roots. As mentioned, the mulberry will be having a taproot system, and the stem it is showing an alternative branching type of the stem, and the leaves, if you see, there are in these leaves they have a good venation that has been observed. Then in this, through this internal anatomy, we will be understanding the distribution of specialized cells called lactifers and idoblast cells. Here we will also analyze the vascular ring organization in the stems. What is vascular ring organization? We have vascular system that will be observing in this unit. What is the functional insight of this lesson? It is we will examine how these internal structures directly support the production of high quality leaves for silkworm. As mentioned, mulberry leaf, mulberry plant will be directly defining and directly affecting the growth and the development of the silks. Here this is a functional insight that we are going to study in this unit. Firstly, we are going to see the internal anatomy of root. Let me tell you, the root system that is present in the mulberry, it is a taproot system. This taproot system the root that is present underground, and it has a primary root, and it gives an anchorage and a good support. I mean, it gives a mechanical support and also a good strength for the plant. Then we'll see the root, the outer layer. The outermost layer of the root is called as epidermis or epilemma. That means the outermost layer, it is a single layered structure which is present. It is a thin walled and it will lack a cuticle to facilitate water absorption. So the major function of the root is the absorption of water, the absorption of minerals, and also giving a good strength and support, good mechanical support for the plant that is above the ground. Here this is the epidermis. If you see the cross section of the root under a microscope, these are the structures this is how the structure of the root that we can see. This is a taproot system type of a modification, and the outer layer, it is called as a epidermis or epilemma. This is a periferous layer. This is called as a periferous layer. It is a very thin walled layer and it has a root hairs that are present. The major root hair, it is a unicellular root hair that has been present. It extends the layer, and the major function of this root hair is also it absorbs the water which is present in the soil, it absorbs the minerals that is required for the plant, and also it is helpful for the increase of the surface area for the nutrient update, for the nutritious levels to be upgraded in the plant. Root also plays a major role. If we see the second layer of the root, it is called as cortex. So if you see, this is the outer layer, and we are seeing the cortex. The cortex, it is having a parenchyma cortex. That means a broad zone of loosely arranged cells. Usually parenchymatic cells, these cells are loosely arranged in the root, and the role of this parenchymatic cells, it will be storing food, and it will allow the water movement to be done in the root. That means as we pour water in the mud, the roots will be absorbing the water. Yes, which cells are allowing the root to absorb the water? Here the parenchyma, which is present in the root cortex, it is helping the plant, the root, to absorb the water. In the same way, this parenchymatic cells, they are loosely arranged. They are not compactly packed, rather it is loosely arranged, and it also helpful for the storage of the food, and also it helps for the movement of the water. The next innermost layer of the cortex, it is called as the endodermis. The innermost layer of the root is called as the endodermis. As this endodermis, it is the innermost cortex layer which will be having a Casparian stripes. This Casparian stripes, it is made up of the suberin. It is made up of the suberin, it will be helpful to regulate the fluid flow. As mentioned, this will be the major function, is it will be helpful for the fluid flow from inside the root to the outside the root. That means traveling and the transportation of the water molecules from the outside to the inside of the root is being done because of the presence of the suberin. That means because of the presence of the Casparian stripes. This is made up of the suberin. If we see a pericycle, it is an uniseriated, it can be this pericycle, it can be a uniseriated layer or else a multiseriated layer, which will be helpful in the development of the lateral root development. This pericycle, if you see the pericycle that is present, that pericycle, it can be one layered or one seriated, or it can be multi seriated. It will be helpful for the internal, or it will be helpful for the lateral root development. If we see the root vascular system, this root vascular system, we know that the vascular system, it is nothing but xylem and the phloem. Xylem will be helpful for the storage of the storage or the transportation of the food materials, and the phloem, it is majorly for the water, for the fluid flow. It can be diarch or in the tetrarch condition. The xylem bundles are in the diarch. Diarch are nothing but 2. Tetrarch nothing but 4. That means in the vascular system, the xylem bundles might be 2. If it is 2, that condition is called as a diarch. If it is 4, it is called as a tetrarch type of a condition. That means in the root vascular system, there are 2 types. One is a xylem and the other one is a phloem. Xylem is responsible for the transportation of the food, and the phloem is responsible for the transportation of the water molecules. And this xylem bundles, it can be 2. If it is 2, that condition of that type of condition is called as a diarch type of a condition, and if it is a 4 vascular xylem bundles are present, it is a tetrarch condition. This xylem and phloem are not arranged in a continual manner in this mulberry. Rather, it is been arranged in an alternative manner, that means in a radial manner. One, if the xylem is present at a place, the next would be a phloem, then a xylem. It is an alternative, it is arranged in an alternative manner. That means one xylem, one phloem, and xylem, phloem. It has been arranged in that type. So this xylem is exarch in condition, meaning the protoxylem, that means the older cells will be towards the periphery and the newer cells will be towards inside. Then we see the secondary thickening layer. It creates a continuous woody core as a root edges, because this the root edge will define the thickening of the latex, or it depends upon the, depending upon the age of the root, it will be, the woody core will be developed. Next we have the microanatomy of the stem. Till now we have studied the root, the outer layer, the middle layer, the innermost layer. Then we have studied the vascular system that is present in the root. Now we are going to study the microanatomy of the stem. So this stem is present above the ground, that means above the soil level. The aerial part of the plant, it is called as a stem. If we see the cross section of the stem under a microscope, this is the view of the stem. If you see this is a young stem, the cross section, it shows a highly organized ring of vascular bundles. It is called as a U-still. Vascular system, it consists of xylem and phloem again, which will be helpful for the transportation of the food and the water. Here if you see each point, each bundle is conjoined, it is collateral and open type. That means it will be containing an open cambium, and it will be allowing this type of condition will be allowing for the growth, for the diameter growth, not through the apical growth or the lengthwise growth, but this system, this U-still ring will be helpful for the diameter growth. If we see the tissue hierarchy in the stem, the first one is a hypodermis. This hypodermis is made up of 3 to 4 layers with a collenchymatic cells. That means the collenchyma cells are present. In the root, the parenchyma cells are present. In the stem, we see collenchyma cells are present. This collenchyma cells will provide mechanical support, and also it will be helpful for the flexibility to the young branches. That means in this mulberry leaves, stems are arranged in the alternate manner. Alternate branching of the stems are being present in this mulberry plant. So this, as it is present in the alternate manner, the mechanical support also is important. Here if you see this hypodermis, this will be made up of 3 to 4 layers, that means multi-layered. It is called as a multi-layered, which is made up of, which is filled with the collenchyma cells. This collenchyma cells will be providing the mechanical support, and also it will be very flexible for the adapting to the young branches as well. if you see the endodermis in the stems, it is often accumulates, it segregates the starch grains that are present, and also it will be acting as a nutrient reservoir during the growth phase. It will be majorly acting as a nutrient reservoir. That means all the nutrients that are be present are been accumulated in the stem system, and it has been present in the starch sheath. Next we have a stem composition. If you see the percentages of the stem composition, the vascular tissue, it is 55%, it has been covered. The cortex, it is 25% of the composition it will be holding, and the pith, that is a central part or the medulla, it will be consisting of 15%, and the epidermis will be consisting of, the composition of it is 5%. So this U-still type of an arrangement, it will ensure the efficient nutrient distribution. As mentioned, the starch sheath that is present in the stem, it accumulates the nutrients, and also it is also called as a nutrient reservoir of the plant. So while maintaining the structural integrity, and also while maintaining the mechanical strength, and also the support that is coming from the root, this stem will be having a U-still type of an arrangement. Then we see a sap system, that is lactiferous. Majorly this mulberry plant comes under a family called as Moriaceae family. So the major characteristics to be identified and to be placed under a Moriaceae family, it is because it will be having a milky latex. That is why this mulberry plant is placed under the family called as Moriaceae, and this mulberry genus of this mulberry is Morus. This is having a milk sap channels. So the specialized feature in the mulberry, it is having a milk sap channels. This is the specialized channel that is present, and it has a specialized cells that is called as lactiferous cells. Throughout the cortex and phloem, this lactiferous cells are present. Throughout the cortex and also throughout the phloem, these lactiferous cells are being present. So this is an unbranched tubes. This will be containing milky latex. It doesn't contain a watery type of a latex, rather it will be consisting a milky latex. So that is why it has been differentiated, and it has been identified and classified under the family called Moriaceae family. Though it comes under a kingdom Plantae, as this is today we are studying about the sericulture botany. So this comes under a family called as a Moriaceae, because of the specialized cells that are present in the cortex and also in the phloem. This is present inside the cortex and also inside the phloem. So this system will be serving as a defensive mechanism against the herbivory and aids in the sealing woods. That means because of the specialized cells that are lactiferous cells, it is helpful for the stem. In the stem of the mulberry, if you see, this stem is regenerative and it grows rapidly. It is regenerative after the pruning. After the pruning is done, it has an ability and it has a capability to regenerate itself, because the cells divide continuously and it has a ability of regeneration. Here in the system, as it is having a specialized cells called lactiferous cells, it helps for the defensive system. It helps it defends itself from the herbivory, and also if it has been injured, or if any infection or any disease has been caused, it can be healed all by itself, rather putting any fertilizer or any medicinal powders or anything on the plant. It has an ability to recover from its own injury, and also it has ability to recover from its own infections that has been caused. So this is about the internal anatomy of this stem. Now we are studying the internal anatomy of a leaf. The engine of the photosynthesis. This is also called as an engine of photosynthesis. As mentioned in the sericulture, the silk industry, the silks will be feeding on the mulberry leaves. These leaves are highly nutritive, and it will be consisting of high protein and high carbohydrate contents as well. So as we see the anatomy of the leaf, This is a dorsivent. If we see the cross section of the leaf under a microscope, this is the structure. This is how the structure can be seen. These are the phloem that are present, and the inside, the towards the inside we see the xylem contents, and the upper, it is the upper layer is called as cuticle. It is covered by the cuticle and the epidermis. If we see, this is the dorsiventral leaf. This mulberry leaf is the dorsiventral, meaning the upper and the lower surfaces have different cellular organization. The upper surface will be completely different, the organizations of the cells will be completely different, and the lower side of the cell of the lower side of the leaf cells arrangement would be completely different. So here if we see in the picture, there are it is a layer. The epidermis is covered by a layer called as a cuticle. This cuticle is a thick waxy type of a cuticle which is present, and it covers the epidermis to prevent excess of transpiration. What is transpiration? Transpiration is nothing but it doesn't allow the leaf to water droplets to be transpired outside, and it doesn't allow the water molecules to move outside and to be transpired. So this cuticle, it is a thick waxy type of a layer, so it is very difficult for the water to be coming outside. So because of the cuticle, the water transport system will be very less. That means the transpiration, the rate of transpiration will be very less due to the presence of the cuticle. Though the epidermis is present, but it doesn't play a role of the transpiration. Rather, the cuticle, it plays an important role. If the cuticle is absent, there in a few structures we see the cuticle will be absent. In such kind of a leaves, in such kind of a condition, what happens? The water transpiration rate will be very high. That means the transpiration will be high. Now if we see the functional cell units, as mentioned, there are specialized cells that are present in the mulberry. First one is a lactiferous cells. Now second one is a idoblast cells. This is a large specialized cells which is present in the epidermis, which will be consisting of the statoliths. Cystoliths. What is cystoliths? It will be consisting of the calcium carbonate crystals. Hence it is called as cystoliths. Because this cystoliths are present, this is this type of a cell is called as idoblast. Then we see stomata. Stomata is also called as a guard cells. It will be having a guard cells are present, and which will be helpful in the outermost layer itself. It will be present, and it is found primarily on the lower epidermis. They are flanked by a kidney shaped guard cells to the control of the gaseous exchange. As mentioned here, the cuticle will be responsible for the water transpiration system, and here in the stomata, gaseous exchange system is been done. That means two guard cells are present in the bean shaped. It will be helpful for the transportation of the gases. Then we have a trichomes, that is a unicellular. As in the root we see a root hair, it is a unicellular root hair that is present in the root. Here in the leaf we see trichomes, that is an extracellular growth that has been done in the leaf. It is also unicellular hair that reduces the transpiration, and it protects the leaf from the insect injury. So in the root we saw that the root hair will be absorbing the water, it will be absorbing the minerals, and also it will be helpful for the water transport system. Here in the leaf, this will be protecting the leaves, this will be protecting the leaf from the transpiration. Though the cuticle is present, it does up 75% of the job, and 25% of the job is done by the trichomes. These trichomes also help the mulberry leaves from the water transpiration, and also we see majority of the mulberry leaves, as the silks will be feeding on the mulberry leaves, we can see different types of infections that are being caused, or else the injuries while harvesting the leaves. What happens? This trichomes, because of the trichomes, what happens? If the injury, even the injury is being caused by any insect, it will be regenerating, or else it will be sealing itself from the injury. Then we see the mesophyll engine. What is mesophyll engine? The middle part. The middle part is called as mesophyll. This mesophyll engine, if we see, which is made up of a palisade parenchyma and also the spongy parenchyma. In the root we see the parenchymatic cell, in the stem we see the collenchymatic cells, in the leaf we see palisade parenchyma and the spongy parenchyma. If we see this palisade parenchyma, this is 1-2 layers which are elongated, which are vertically packed cells which are rich in chloroplast, which is located just below the upper epidermis. That means here it is just present beneath the upper epidermis, the mesophyll, the middle layer that is present. It is a vertically placed. These cells are called as a palisade parenchyma. Now these not one layer but 2 to 3 layers are present, and majorly it is rich in chloroplast. Chloroplast, it is a pigment which gives the leaf green in color. So this is just this palisade parenchyma, as this is a cuticle, the outer covering. This is the first layer, it is called as an epidermis. This long elongated cells that are present, these cells are called as a palisade parenchyma, which is also helpful for the which also gives a mechanical support for the leaf, and also it will be highly consisting of the chloroplast. Then we see a spongy parenchyma. These spongy parenchyma, they are not compactly packed, but it is loosely packed, many air spaces. It is consisting of many air spaces. Because of the air spaces that are present in the leaf, it will be responsible for the gas circulation for the photosynthesis process. If you see here, this is the palisade parenchyma, and the loosely arranged cells, if you see here and there dispersed cells that are present, it is called as a spongy cells, which will be helpful for the gaseous exchange of the in the leaves. In sericulture fact, if we see, higher palisade to spongy ratios are assisted with better leaf quality for the silkworms. Here I can say what is important in the sericulture, it means if the cells are arranged from the palisade parenchyma to the spongy parenchyma, that means the gaseous exchange are well done. If the leaf is well developed, if the leaf is mature, if the leaf is correctly arranged, it will reflect the quality of the silk. It will affect the quality of the cocoon as well. Here if we see, the major focus of this taxonomy of mulberry, it is cystoliths. Cystoliths, it is a specialized cells I have mentioned, because it will consist of the calcium carbonates. What is the role of this mineral storage? What is the role of this mineral storage is, cystoliths are stone cells. These are also called as a stone cells. This is made up of a calcium carbonate. This cystoliths are made up of the calcium carbonate. They will be helping and they will be serving as a metabolic reservoir, as they are help in the deterring of the leaf eating pest. If you see, majorly silks will be feeding on the leaf. Majorly the leaves might sometimes get damaged, or sometimes it will tear off, even though this mineral storage will be present. So they are key botanical feature of the Muriaceae family. As mentioned earlier, this mulberry comes under a family called Muriaceae because of the lactiferous cells that are present, and also cystoliths that are present, because of which this is been differentiated and placed under a different family called as Muriaceae and under a genus. Then we have organ comparison, internal organ comparison. If we see the vascular order, it is the root, stem and the leaf. As we have mentioned, the root, the stem and the leaf, the vascular system is present, and this is a radial or exarch type of the condition which is seen in the root. In the stem we see the collateral or endarch type of condition, and in leaves we see bundles, that bundles in the midrib, that is a major rib, major vein is in the leaf is a midrib. Then endodermis, that means in innermost layer of the epidermis is called as endodermis. Here in the root we see the Casparian stripes. This Casparian stripes, it is made up of the suberin. As it is made up of the suberin, the water transportation will be very easy in the root. That means water transportation, the water flow or the liquid flow will be easily because of the Casparian stripes which are present in the root. Next we have a starch sheath which is present in the stem. This starch sheath is also called as a nutrient reservoir of the stem, because it will be storing all the nutrients, and it will be consisting of the xylem and the phloem. If you see in the leaf bundle, if you see in the leaf, it will be having the bundle sheath. That will be helpful for the water transportation again, and also protection, regeneration of the leaf. There are no specialized cells that are present in the root. It is a simple taproot type of a system. And in the stem, if you see, lactiferous are present, and lactiferous nothing but a milky waxy type of a structure which are present in the stem. If we break the stem, we see the milky type of a consistent liquid that will be coming out. That is called as a lactiferous because of the presence of the lactiferous cells. Then we have a leaf. In the leaf we see a specialized cells called as idoblast. This idoblast, why is it called as idoblast? Because of the presence of cystoliths. This cystoliths, it is made up of calcium carbonate. We see the ground tissue. If we see the ground tissue, this is a broad cortex. This stem will be having a cortex and the pith. Then we see this leaf. In the leaf we'll be seeing it will be filled with the mesophyll. That means this mesophyll will be having a palisade parenchyma, and also this leaf will be having a spongy parenchyma. Palisade parenchyma and spongy parenchyma will be reflecting the health of a leaf. Next we see the holistic botanical strategy. The holistic botanical strategy of the sericulture means, here in this taxonomy we see macromorphology, that means the outer structures, stem, leaf, outer structures, and the internal anatomy as well. Internal anatomy is nothing but the tissues that are present inside the leaf, inside the stem, and also inside the root. They all work together. That means the coordination of the macromorphology and also the internal anatomy will be reflecting the health of the mulberry plant. The deep root system will support the woody ustial stem, which carries nutrients to the idoblast rich leaf. That means if the root is strong, that means the stem will be strong and the leaves will be healthy. That is how the mulberry plant are being interrelated, as the coordination and the correlation between the taproot, between the root, between the stem, and between the leaf reflects the health of the mulberry plant. So the major success in sericulture, it relies on managing these biological features through proper soil care and also through the proper pruning. How can I say proper pruning? Because depending upon the conditions, you can manage the plant, whether to keep it as a shrub, whether it to be kept as a bushes, whether it to be kept as a large tree, depending upon the climatical condition and depending upon the habitat and depending upon the soil type, the mulberry plant has to be taken care of, because of which the whole sericulture, the silk industry will be improvising, and by educating farmers on all the above features and all the above studies will improvise the silk industry and the sericulture, and it gives the scientific knowledge to the farmer as well as to the science department to improvise and to in the advancement of the technology.